After that, Ann, would you introduce me in Wyoming and California too in the upcoming weeks? Hi, everybody. Ann didn't tell you that I lived in your area here in Bend for almost 10 years. And I've been in Oregon for about 40 years. Oh, my God. And did my career here, raised the family and all that good stuff. So I'm at home here with you, and I know a lot about the wonderful things that are going on in Bend area during this summer. I had an experience about 3 days ago that I'm just burning to share with you. It is just so fortuitous. I was in the gym in Ashland, and I usually go to the spa to kind of relax after doing my little weight deal and all of that good stuff. And there was a guy in the spa, I hadn't seen him before, you know, and we just got to talking. Well, he was from France. And, you know, these days I can hardly have a conversation with anyone but getting to the genetically engineered organism concept. And he said, I'm an American citizen now, and I know a lot about your Constitution. I said, well, what's going on in France? He said, well, I have a dear friend who's been in jail twice because he's an activist. Now, that took place a while back, Ray, about 10, 12 years ago, when, you know, your company, Monsanto, was trying to get our farmers to grow corn. And we were concerned about the cross-pollination. Oh my God, I'm thinking 12 years ago, this guy's ahead of the researchers in the US. And I said, well, what happened to him? Oh, after a while, you know, they released him when things quieted down. But he says, you know, Ray, I had to study your Constitution to become a citizen of this country. And your First Amendment says something like freedom of speech and expression, including writing. And I read that about 90% of the American population wants to have labeling on their foods. What's wrong with your government? What happened? Tell me about that. You know, this really happened, right? I just had to share that information with you. Something else that I need to share. Do you know that approximately 1/3 of the people that are alive on planet Earth, about 2 billion, very likely know more about their food than most Americans? How does that correct? Does it make you angry? Does it make you, what the heck is that old guy talking about? Come on. How can they know more about their food than most Americans? Well, these are the people whose countries, whole or in part, demand a label on their foods. And that label simply says, contains GMO-derived material or something to that effect. Or it might say GMO-free. So I ask you, why are we even here today? Why do we have to complain about this? When people from Europe, you know, have these kinds of conversations, when 2 billion people see the label on their food, do you know it could be less cost for industry not to have to segregate American corn that's going to Europe and they have to put a stamp on it, and they segregate what we get without the stamp. Let's not bother with the segregation. Let's just put the stamp on everything and call it what it is. That should cost less money, right? But that's not what you're going to hear about 6 weeks out from the election. You know, for the longest time, I talked as an independent person down in Jackson County. I thought, you know, maybe it would be more credible with people like yourself if they knew that I wasn't part of the campaign. I wasn't a politician. Well, I'm not part of this campaign. I don't have any inside information. Ann and her colleagues that help support this event today, they don't know what I'm going to say. I hope that they appreciate, and I hope that they like what I say, and you too. but I'm speaking to you from my heart, from my experience as a scientist. And you're going to hear today about what I like to call the lemming effect. You know, these are these little animals when they get spooked. They run and run and run. If they come to the edge of the cliff, doesn't matter. The first one goes off, then the next one goes off, and pretty soon most of the herd goes off the edge of the cliff. Well, that's what's happened to Americans and to politicians who think they know what there is to know about genetically engineered crops. I'm sorry, they don't. They don't. And I haven't figured out why they haven't asked me. But they haven't. Okay. In Southern Oregon, since the election in May, we have labeling already. That's the purpose of this slide. This is Josephine County, and by the way, they had their election win by the 16% spread. I think it was 58 to 42 on a budget of $30,000. Isn't that fantastic? Jackson County, the dear leader of our group in Jackson County, I love this phrase. She said, let's use the phrase, you can't buy Jack for $1 million. Because that's what the opponents put in to ours, I don't know, $300 and some odd thousand. And we only won, as Ann said, by about the 66 to 34%. And I'll tell you folks, this is not a partisan situation. And it shouldn't be and it can't be. Jackson County, Josephine County have more registered Republicans than Democrats. And we are so proud, all of us, that we got a handful, hundreds of local businesses and local farmers to publicly support the banning of the planting of GM or genetically engineered organisms. Farmers need to be included in this discussion. This is what a label might look like. I pulled this off the internet. Some of you may have seen this, non-GMO. This contains genetically modified, so it's nothing fancy, nothing sophisticated, a little bit of ink and a little bit of a rubber stamp. So today I'm going to change the pace as I go through. Some of it will be slower than faster. If we get a little bit long and if people start nodding off, I'll probably jump up and down and skip a few slides, so bear with me. But we're going to try to talk about just what is this technology? How are these things made? How are the crops made, genetically engineered, you know, stuff that we eat? We'll talk a little bit about labeling, but not a lot. And the promises and the propaganda, is my terminology, that has resulted in the incredible worldwide proliferation of the technology. And then I'm going to share with you my concerns about the ecological ramifications of the management of these crops. So, terminology. These are all the same things. A recombinant organism, a transgenic organism, a genetically modified, or a genetically engineered. Most of the time in the press you'll see GMO. That's what you're used to. But scientists like the term transgenic. And that's from northern Mexico to the southern tip of the Western Hemisphere. That's what our Latin neighbors use. They're transgenicals, not GMOs. So, scientific deal, a little cultural deal. Okay, all of this technology started in the 1980s and field releases in the 1990s. With a bunch of promises from industry. For farmers, increased crop yields, more money. 70% of the farmers like that concept. Decreased use of pesticides. Convenience of farming practices, and I'll show you why. Environmentally friendly, use of less fertilizer. Wow, as good as sliced bread and then some. Now, how many of these promises have been fulfilled? And I'm not going to answer that rhetorical question. I'll present the information to you that will allow you to answer it. First of all, we're going to have a little fun with Ray's demo with the ropes. But the process involves something that happens in the laboratory, and it typically merges a few genes from typically very different organisms, giving forth to unique, novel, one-of-a-kind combinations of genes that ain't going to be found naturally anywhere on this planet, don't happen by conventional crossbreeding, and unlike plant cross-pollination, this is an asexual process. But you will be told that this is just like natural reproduction and it's been going on for 10,000 years. And this is not really what I'm talking about. Shooting a fish to get a genetic change in a tomato. Here's what I'm talking about. I'm going to put the mic down for a moment so I can do my little demo along with the slides. So, what you've got here are actual just incredibly magnified pictures of actual DNA molecules. And can you all hear me okay at this moment? Okay. So, if this is meant to be a bacterial chromosome, think about it having 1,000 segments. And each one of those segments is a gene. That's about what your typical bacterial cell has. And most of our crops that are genetically engineered have 1, 2, or 3 genes taken from a soil bacterium and put into the plant. In some cases, a gene from a plant virus is used to change the characteristic properties of a plant. So the object is to take in the laboratory what we call a scissors. Let this be our DNA molecule or chromosome from a bacterium, and we'll take a scissors and cut out the genes that we want from the bacterial chromosome. And maybe we'll do 1 chromosome from 1 bacterial species, or 2, or a plant virus, but it's all the same concept. Maybe we want this gene that maybe is responsible for making a toxin we call Bt toxin that kills insects. So maybe that's what we'll want. So we cut those out. And here, let this represent the 3 individual genes. This one, let's say it confers resistance to the herbicide called Roundup. This one makes the toxin that will kill an insect that starts chewing on the plant. And this may come from a virus, and we'll call this like the promoter or the orchestra leader that affects the action and activity of these 2 genes. So they all 3 need to be there. They all will cooperate and work together. So the question then becomes, we've got these genes from bacteria, how do we get it into the plant? We use another type of DNA molecule for that called a plasmid. These are not exactly to scale. A plasmid, DNA plasmid is about 1, 2, or 3% the size of a bacterial chromosome. You don't know it, but you know all about bacterial plasmids because that's what confers antibiotic resistance to pathogenic bacteria, is the data that's carried on a small DNA molecule called a DNA plasmid. So because it's tough, it's easy to handle, change, manipulate in the laboratory, scientists work with the DNA plasmid and they cut it open the same way as they cut the chromosome open. Everything gets put into a single test tube, and lo and behold, these genes rearrange themselves, come back together, and will form a new type of DNA plasmid called a recombinant DNA molecule. Recombine genes from bacteria, and then this is the molecule, this is the DNA that gets sent into the genetic material of the plant. How is that done? This new small circular DNA molecule gets mixed with bullets Not fooling you. Bullets. And those bullets are microscopic particles of gold. And the DNA sticks to those microscopic particles of gold. That material is then loaded into a gun. Here's the gun, right here. That gun contains then these microscopic molecules of DNA plasmid attached to a piece of gold. We then tear apart, shred up some plant leaf, and use the gun to shoot this material into the nucleus, to set apart the genetic material of individual single plant cells. Those individual plant cells then are placed onto a nutritive medium in the light and asked to regenerate themselves, regenerate little baby plants from one cell. And this is what it would look like. There are the little plantlets. Now, you're looking and you're seeing some of them are white, some of them are green. Not all of the cells that get shot with the gold bullets integrate or incorporate this DNA into the nucleus. In fact, it's a low percentage. DL, so far as we know, it's done more or less randomly. So we have to set aside and separate the cells that worked, that received the toxin gene, that received the resistance to Roundup, that have the virus DNA to help everything replicate, from all the rest. So it's really an easy process. Once we get it to this stage, we have these baby plantlets, we can just spray this plate with some herbicide, Roundup. And the little plantlets that live, as shown on the right, are Roundup resistant. That's why they survived the spray. The ones on the left are dying. That's why they're white. They're checking out. They're still sensitive to Roundup. So these little plantlets, now that have leaves and a residual root, can be put into soil and regenerated into a plant. The properties of this plant might be now Roundup resistance. And by the way, you might hear me say glyphosate. Glyphosate is the active ingredient in Roundup. So if I mix up glyphosate, Roundup, forgive me, I'm still talking about the same chemical. So this plant may, as I just said, be Roundup resistant. It may also express the insect toxin capability. And that is screened by adding insects to this plant. They'll eat the leaves, the insects die. Voila, that's what you want if you're an industry. Now, who does this kind of work? Where in the world is this research being done? Outside academia. Where is it being done? It's commercialized, right? Farmers are buying these genetically engineered seeds. Who's doing this work? Anybody know?
OSU.
Chemical companies. Chemical companies. What's a chemical company? Oh, those are the folks that starting in the 1930s brought us the miracle chemicals such as DDT, PCBs, later Agent Orange, later 2,4-D, later insecticides that are killing honeybees, and now, today, Roundup. What happened? Why would a chemical company, internationally known, and there are about 5 big ones, want to integrate themselves into our food system? How does that work? Well, it works because if you're a farmer and you buy their genetically engineered corn, you sign a contract, and by the way, you don't really buy it. You lease it. You lease those seeds. You sign a contract that says, you know that this seed is resistant to Roundup, the plant is. And we want you to buy some Roundup from this company, chemical company, so you can spray it on our crop, our seeds that you have leased from us for one crop year. And we're going to suggest you spray 3 times, whether you need it or not. Make sure, kill all of those bad weeds 3 times a year. And by the way, we have developed a technology, in addition to the genetic technology we've just shared with you, changing the genes in the plant. We can also send you some more seeds, really, really special seeds that are coated with a talcum powder and colored blue, green, pink, that bring with it 5 more chemicals. You need it because it will help you in controlling your pests. So you get the story. Chemical companies have inserted themselves into our food system. And what a business plan. These are the vehicles now for the chemicals that they sell. It's our food. And hopefully it's safe, right? Because we have regulators in this country that look after that process. I was in the research division, by the way, of EPA, not the regulatory division. Can you tell? Okay. You will hear the other side say, the process that I just shared with you has been going on for 10,000 years. No, I'm sorry. This is the process that's been going on for 10,000 years. It's called cross-pollination and hybrid selection and hybrid vigor, not the asexual genetic engineering. Humans have been genetically selecting for more vigorous plants, corn and soy, etc., for 10,000 years, but not using genetic engineering techniques. Where are these things found? You know, cotton, canola, soy, corn, of course, and all of those products, sugar beets, a little papaya, and some zucchini squash. They are found in our junk food, snack food, which I eat, but you know, there's a whole list of products that are not genetically engineered, snacks that compete. Food. That's part of the farm program that was started in very serious fashion in 2005, in which farmers in this country were given an incentive to produce corn for ethanol production. And that ethanol then is put into our gasoline to help us become a little bit more energy independent. So 45% of the corn that's grown goes into that process. 45% of it goes to feed our livestock. That leaves about 10% left. And you know where that 10% goes? Most of it? That's the seed for next year. Only 10%. Oh yes, and it is also approximately what goes into our snack food. 10%. So 45% ethanol, 10% for you and I. What a deal. Alfalfa was approved just about 2.5 years ago. And anybody that is into health foods like alfalfa sprouts, good luck with that. It's not going to be labeled genetically engineered unless you buy it from a specialty store, and then it might be labeled organic. I have stood this last spring with a GMO advocate from Oregon State University, right here to here, and he used this slide to say, and you see the incredible explosion of this technology worldwide. In 16 years, the land mass planted to genetically engineer crops is the equivalent to 7 states of Oregon. Therefore, it's safe. Huh? Just because it grew faster than any other known human technology in the history of the planet. Safe? Just because it grew. Okay. Well, I said, well, you know, production of polychlorinated phenols. My God, 700,000 tons produced since the 20s, the good old days. Does that mean this massive production, that's also safe? I don't think so. By the way, 90% of it stayed in the US, and about 50% was still in use when this EPA data was finished in 1976, and you don't want to know, and I don't want to know if it's still out there. Okay, these seeds, because they're genetic engineering, I told you they're unique and novel, are of course patented. I don't have a problem with patents. That's part of the American way. You protect your inventions, and no problem. But it can be abused. It's possible. And what am I talking about here when seeds are genetically engineered? This slide I borrowed from a young lady who's an attorney in Eugene, Melissa Wisherath, and she's involved in the Oregon GMO labeling campaign. But the patents on these seeds tell the farmers, you've just leased some material from us. Here's a bag that has genetically engineered corn. You know, it says DeKalb on it, but it's from Monsanto. And if you read on the back, basically it says, when you open this bag, you agree to all of the terms and limitations of this product. In effect, you've signed the lease that says you're not going to contaminate your neighbor. You're responsible for that, by the way. You can't save back any seeds to replant next year. You have to sell all of that seed and make sure that you guarantee the integrity of that stuff. And oh yeah, as I told you earlier, you got to use our chemicals to control your weeds and, if necessary, insecticides. You can't, if you've got a Monsanto lease, you can't buy your chemicals from Dow or Syngenta or a couple of companies in Europe. You've got to buy it from us at Monsanto. That's part of your contract. You know, save the seeds. You've leased them. The lease is good for 1 year. That's what those patents brought. Okay, a little bit of labeling fun coming up now, and I'm curious at the end, you know, how does this make you feel? So our labeling in the US of our foods is voluntary, and it's all about, you know, if you've got two foods that are very similar, how different do they have to be before you put different labels on them? Well, they have to differ in a way that can be sensed. Size difference, color difference, taste difference, smell difference, texture difference, then this food needs to have a different label than this, even though they may be the same thing. But they really can be sensed to be different. You got to label it. And that's what happened in 1992 when Congress gave the FDA the Federal Food, Drug, and Cosmetic Act. Well, let's see how this works. Let's look at some Atlantic salmon, which if you go to a box store, you've seen. Now, this is the wild-caught, non-genetically engineered Atlantic salmon. That's how it looks. This is the same fish, non-genetically engineered, after color has been added to the food. It's a little bit more attractive for me at the bottom. No genetic engineering here, right? Just some color, natural color, went in with the food. Farm raised, color added, and where's the label? Right here. Color added through feed. It says steelhead, but it could be salmon as well. All right, here's a genetically engineered, same thing, same species, Atlantic salmon. And it has a super hyped up growth hormone in it so that it grows about almost three times faster than its non-genetically engineered component. So the last time that I checked, when something is about 3 times larger than this other thing, I can sense a difference in size, right? Why? Do you think it'll be labeled? Well, what we're asking for is on this sign, in addition to color added, Why couldn't it just say contains genetically engineered products or something like that? We can see how big those fillets are. Okay, here's the bad news. There's now a bill in the pending, it's being developed in the United States Congress to require a national genetically engineered food labeling program. So do you think us Oregonians can just pack it up and go home? We'll love it, right? You know what that label is going to be? It's up there on the screen. That's what it's going to say. 100% natural. And in my way of thinking, it should be 100% unnatural. This is not a natural process. That's what we got coming if it goes anyplace in the U.S. House of Representatives. I talked to you about this, so we'll just kind of skip over this. This represents the list of the 64 countries that have labels for their genetically engineered crops. And when you hear in October that you don't want to pay more for your food, it's going to cost you more to label this stuff. Call up a friend in Vietnam, Germany, Great Britain, China, Russia, South Korea, maybe in South America, and ask them, did your food prices go up when you started the labeling program? I haven't found any information that, if Europeans complain about something, especially if it's costed their food, we usually hear about it because we've either caused it or the problem is coming our way. I haven't heard anything. And, someone has a concern, I don't want to vote for that labeling thing, it's going to cost me more. Well, ask, where did you hear that? Why do you say that? Why do you believe that? Did you get it from a credible source? Did you ask these people, how do they know that? And if they're really candid, they'll tell you it's propaganda. They heard it on a radio ad, or they saw it on a TV ad. One of the reasons about a month and a half, 2 months ago, that I came out as an actual supporter of the ban the planting of genetically engineered crops in Jackson County was that, excuse my expression, the damn lies that came out in the ads. And me being a scientist, that's how I knew these were lies. Technical things they were trying to scare the people in Jackson County that I know wouldn't happen. One was, do you want live in a county where you can't grow strawberries? I swear to God, that was an ad. You won't be able to plant strawberries because they're genetically engineered. They're not genetically engineered. Using recombinant DNA technology. And that's what the phraseology was in our bill. No, you can't afford the cost that'll come your way. What cost? Well, it's going to take away the funding from the libraries. Folks, one person, one telephone call that I made to the county commissioner's office, and I asked, I don't know if you've seen these ads. Can the enforcement of this bill actually dig into the pockets of the library fund and take money away? No, that's a separate pot of money, can't be done. 2 examples of the lies and many more that will come. Not my opinion, science opinion, factual opinion, county commissioner opinion. So I have 2 slides that I want to quickly share with you, show you exactly the difference coming your way between commercial farming using Roundup Ready crop system and the old-fashioned conventional farming. Here we have conventional. It's a commercial cornfield, and the ground has been tilled before the seeds are planted. Clean, no weeds except, here are a few. Because there is a chemical that's put into the soil at the time the ground is tilled. It's called pre-emergent. And as soon as a weed seed begins to germinate under the ground, it gets zapped. No spraying after the crops are up because the crops will die. They're sensitive to Roundup, right? So this farmer either can go out and hand spray a few weeds, or he can hire day workers to go out and pull them. Or if he's really, or she has really done their job, it will be pretty much a weed-free field like this and be ignored. Let's take a look now at a comparable Roundup Ready plot. And I'm going to come back. Remember, this is tilled. No genetic engineering. The soil is disturbed. The farmer's got to go over. He's got to spend his diesel. He's got to spend his time. He's got to work the soil. The big advantage that is touted. Here is a field of genetically engineered soybeans. And this was sprayed 2 days before the photograph. That's why the grass, it looks like a pasture field. There's been no maintenance done on that. So he's either gone over it with a boom or a helicopter, and everything is sprayed, right? The crop is sprayed, the weeds are sprayed, everything is on its way out that isn't genetically engineered Roundup resistant. This is no-till. That's why everything is so dirty. There's a lot of weeds growing here. Let's go back now to this same field 2 weeks after this picture was taken. And I'm going to try to illustrate 2 or 3 things at once in this slide. Okay, there's the grass. It's deader than a doornail. There's our monarch butterfly that says, help me. Help me because the weeds that were sprayed are milkweeds. They're gone. So, this beautiful creature has no place to lay eggs and to grow the larvae. And what's happened in the 17 years of Roundup-resistant crops, you know, these beautiful critters fly all the way down to Mexico to overwinter in the high mountain forests. They hang on to the trunks of trees in the winter months, and they occupied a space that was about 45 acres of forest in 1995. In 2013, it's down to 1.6 acres. Now, I'm not going to show you all of the other data that verifies the cause and effect, but it is Roundup, and it is the management system at play, and it is the removal of over 60, 65, 70% of the milkweed from the Midwest that caused this decline. The other thing that I'm showing you in this slide, note the arrows. The tips of those arrows are pointing to little tiny, tiny, tiny plants that have survived all the spray. There's 1 blade of grass and there's 2 broadleaf plants, and I hope you can make that out. About a month from now, these little green things are going to be called the Roundup-resistant plants. And this is what, on a worst-case basis, not the same field, but a worst-case basis of a soybean field in Illinois, what it looks like, what it can look like. This is weed, horseweed, growing up, taken over a field that was planted to Roundup-resistant soy. The black stalks are the stalks that are still sensitive to Roundup. The green have survived the spray. It's called evolution, and it's called, you have selected now for weeds that are resistant to the herbicide Roundup. What are we going to do? What is this farmer going to do? He's got to go use another chemical. And now you know he can't use another chemical because his crop is only resistant to Roundup. But the chemical industry now has an answer. They have made our crops resist other chemicals by putting in other genes from other bacteria. So what's coming our way, unless the grassroots movement of the United States citizens can stop it, soon we will have 2,4-D resistant plants. We will have them resistant to a chemical you probably never heard of. It's called isoxaflutol. And my dear colleagues in Washington, D.C. EPA say that is a probable carcinogen. And you think, oh, don't worry me. There are carcinogens everywhere. But remember, this stuff is going on your food crops. It's not being hand sprayed in between the rows, on your food crops. And guess what? Even though Roundup may not work anymore, Monsanto still wants you to use it, just in case. So they're not saying use 2,4-D and don't use Roundup. They're saying you better buy both to be safe. Guess who benefits from that arrangement? This is the management system of our Roundup Ready crops. Okay, so we've talked a little bit about the patent restrictions, the labeling, the field management, weed resistance, and the monarchs. Let's go on to the big issue. You know, I just don't want to vote for labeling because I fear it might impact the hungry and needy of the world. Okay, what makes you think that? Oh, you know what? You heard the propaganda put out 20 years ago by industry that promised this new revolution in technology was going to feed the hungry of the world. Right? You know that. You heard that. What am I going to tell you today? Well, I'm going to tell you today that if there's something special about the Roundup Ready crops to feed the world in 17 years, it hasn't happened yet. They're still hungry. They're still waiting. And why? Well, among other things, thank you, Ann, there's no differences in the yield. There's no differences in the yield between genetically engineered crops and conventional crops. Do you believe that? Because you sure have been told otherwise. Let's quickly take a look at the facts. I'm going to show you 3 slides. This is from a corn growing contest, annual contest in Ohio, where they really know how to grow their corn. 19 varieties of corn were included in that test. 16 of those varieties were genetically engineered, 3 were not. This is a contest done throughout the state of Ohio. And there are field observers that go out to make sure everybody's playing by the same rules. And what they found was 219 bushels of corn per acre was the average for 11 and 12. You know what the average is for the total United States is 160 bushels. My God, here in Ohio there are 219. My God, those farmers know how to do it. The non-genetically engineered 3 varieties averaged throughout the state about 221 bushels. And the genetically engineering, 219. Now, that doesn't make this better. They're so close, they're equivalent. Mathematically, statistically, they're the same. But in full disclosure, I will tell you that there are 6 varieties of genetically engineered crops that did outperform. 6 varieties of the 19 of the genetically engineered crops outperformed. Well, that leaves 10 that didn't. So the bottom line is equivalence. Let's go to my friend's data. This is from Professor Jack Heinemann. He's at Christchurch University in New Zealand, and he compared corn yields throughout the United States with that in countries in Western Europe. Whoops. So this is time, and this is the amount, a weight, a metric measure of the weight of corn. And over time you see that corn yields increased. They increased in Europe, they increased in the United States. The U.S. line is dashed, and you can see that about 1995, 1999, we gave up, and we're all about the same yield. Same yield, but a little bit difference here. In Europe, it's non-genetically engineered. Same. It's the same. And I can show you similar graphs for soybeans. But for me, my aha moment after talking about this for a year, a few months ago, the USDA listed a report. Over the first 15 years of commercial use, GE seeds have not been shown to definitively increase yield potentials. In fact, the yields of the herbicide-tolerant or insect-resistant seeds, that's Bt toxin, may be occasionally lower. That's what I just said. It all washes out. Roundup resistance doesn't tell you anything about I'm going to make more of it. The Bt toxin gene has nothing to do with yield. Why should it be better? Not necessarily worse, but why should it be better? It also has nothing to do with the promises of growing in highly saline soil. It has nothing to do with the promises to withstand drought. None of that. That's all propaganda. Wishful thinking in the mill. It hasn't happened yet. Okay. I want to quickly talk about something that happened in the gasohol production program, the alcohol from corn program. What this means is farmers were given this great incentive by Congress to plant more corn, and of course it's genetically engineered corn. And the little starch granule in that corn seed would go to make ethanol. And they had to rip the hell out of over 3 million acres of virgin prairie land that's basically now all gone in the United States. They had to drain those ponds. They had to fill those ponds in, till it, plant it to corn, and generate corn to go into the gasohol program. Remove all of the water and you go to this. What happens to the aquifer recharge into the ground? And does this increase the risks of flooding? You don't have any place to park all of the rainwater or the snow melt. Oh, look at this. Now, I cannot prove to you that this horrendous flood along the Missouri River, where this land conversion took place, is due to taking out all of those ponds. I don't know that. But this flood happened right at the end of the conversion, I believe in 2011. And the extent of the flooding was 50% deeper water than ever recorded in history along the Missouri River. And it flooded in 13, and it's flooding again this year, but not in 12. 12 was a big drought year. One thing we do know, look at that benefit curve. Before genetically engineered corn to alcohol was about a $10 billion a year industry in the United States. Seed plus crop value. 2 years, up to $40 billion. Who benefits from that? Well, I tried to put some of my estimates, and between the seed revenue coming to Monsanto and the Roundup that they sold, they own roughly 30% of that $40 billion. Not too bad of an increase in profits in 2 years. That lobbying of the United States Congress by the chemical industry sure did pay off, didn't it? They're friendly to the environment. Now our beautiful breadbasket Midwest is over 90% genetically engineered crops. So, has anyone in here heard about the dead zone in the Gulf of Mexico? Why? Okay, basically, look at this. It is a heart, and these to me are like arteries. And the arteries here are carrying the water to the Gulf of Mexico. And when that water is polluted, the dead zone in the Gulf of Mexico increases. But now that we have 90% of genetically engineered crops, don't worry, less pollution, not a problem. Baloney propaganda. It's really sad. It's more propaganda. The USDA has said no-till benefits, we're going back to those slides now, right? No-till Roundup-resistant crops. No-till. The benefits of that may be offset by increased crop protection costs, aka you need more chemicals. Yay for the chemical industry. And for some technical reason, when you have all of that dead grass and weeds sitting on top of the ground, the fertilizer does not penetrate to the roots as easily on your plants if compared to when your ground is tilled. Fertilizer gets in. So you got to add more fertilizer. And the fertilizers that you add are required, according to the USDA, to attain optimal yields. AKA, so that your yield with your genetically engineered corn becomes equivalent to what it is for the non-genetically engineered corn. That's what means optimal. Got it? Okay, on the left is a satellite view with the algal bloom that happens in the Gulf of Mexico. The right side is a satellite photograph. The red is the dead zone where there's no more oxygen in the water because of the decomposition of the algae. And the reason why this is serious, there are people that make their living crabbing and shrimping. And those part of the season, those fisheries are shut down. Sports fishermen have to go out further into the Gulf to find fish. More fertilizer isn't necessary. Studies, look at this, non-genetically engineered crops, France, Germany, United Kingdom, and Japan use less fertilizer over time, less over the years, less over the years, less, and the non-genetically engineered crops still yield with declining use of those fertilizers. Ours, we're staying steady to our credit, and not increasing like in India and China, staying level but not going down. So yields, they're equivalent. What's happening in the Missouri River Basin for the gasohol program, we talked about excess fertilizer use and increasing the size of the dead zone. And I think probably should be my last topic. Isn't that beautiful? Those are genetically engineered seeds. I think I showed you that before. They're colored because it's meant to be a warning signal. A warning that these crops are contaminated now with up to 5 chemicals. What chemicals are we talking about and why is it important? Well, they're called cocktails. Thank you, Julia. And most of the chemicals that are added in the talcum powder around those seeds are neonicotinoids, what Dr. Liu at Harvard University in the last week in his national interview called the new DDT. So you can't buy corn anymore that's yellow if you're buying genetically engineered corn. You gotta buy it with more chemicals coming. Cocktails of chemicals. The cocktails will have 1, 2, at least 3 neonicotinoids and an organophosphate. Those are all neurological toxins, all of them. We hope specific just for insects that cause damage. But unfortunately, that's not the case. Colony collapse disorder, the sad disappearance of honeybees throughout the world, has been shown by almost 800 technical scientific reports over the last 20 years. How many do we need? How many scientists have to publish their research results at a Germany, at a France, at a Great Britain, at a Harvard, and so on, to say we know enough, we know enough, we know enough. The neonicotinoids are killing the bees, the pollinators. They're supposed to be killing pest insects on the roots of the plant. To me, it's amazing. The little powder that's on the outside of those genetically engineered seeds, when that seed germinates, the chemicals get absorbed right into the root of the little plantlet. And then as the plant grows, those chemicals go out what's called systemically throughout the entire plant. And as shown here, bees visit corn. They're not in the act of pollinating, technically speaking, but they're in the act of collecting pollen to bring back to the hive. And the concentration of these neurotoxins in that pollen, 4 to 88 parts per billion, minuscule, is more than enough at the high end to kill a bee upon contact. And this is what's being brought back to the hive to feed the next generation of bees. And I thought, just to kind of break the ice, I'd show you this one graph. What this graph is all about is a radar tracking. I mean, scientists go crazy. They put chips on bees and then exposed half of them to the chemical, half of them to sugar water alone, and then by radar, we're able to track their movement back to the hive. So although these French scientists looked at 100 controls and 100 experimental exposures, I'm going to show you just one control and one experimental exposure. So they did this study in 2011 and 12. So let's look at 2012. A bee was fed some sugar water and then released to go back to the hive. And if you follow the green line, that's the control bee. And we can laugh, but I think I now know what a beeline is. Right? You see this road going across the field? That's the line the bees follow to get back to the hive. That's the beeline. And the exposed bee, exposed to the neonicotinoid at a concentration below lethality. Look at this baby. Sad. It goes back to the beeline, but goes beyond it. And I'm thinking, allow me to say things like, it senses it's gone beyond the beeline, and it comes back, finds the beeline, and sadly goes away from the hive in the wrong direction. And I think it senses that it's gone far enough. I have. Sorry. It's gone far enough. It turns around and it's no longer on the beeline, and it just zip, 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 and that's the end of the road. This is pretty conclusive to me that of a neurological reaction in these bees at part per billion levels. And there are other studies that we won't go in today that really directly pin down the cause and effect. And, you know, bless his heart, a week ago, President Obama signs an executive order to study this, bringing together the department heads of about a dozen agencies. What's going on? But damn it all to hell, we already have 800 papers. We don't need any further study. I'm sorry. Color of this stuff doesn't prevent exposure of kids. But I want to tell you that if you look at the eraser on this pencil, if you had the talcum powder covered to that size of that eraser with the chemicals, you'd have between 50 and 100,000 lethal doses of bees. But there are pounds of this chemical put out per acre of farm. What can you do about it? If you know, we all have gardens, I think. We all have insect pests, and I'm sure we've sprayed. But the neonicotinoids come in green and blue bottles, and they will not be labeled as neonicotinoids. They will be labeled by their technical scientific name. So think green, think blue, think death to bees.